Hey folks, and welcome to another edition of Zog Science. Today we're going to be taking a look at viruses. So, what is a virus? Well, viruses are infectious particles, and they consist of two main things: genes, either in the form of DNA or RNA, and a protein coat. Now, some of them can have a lipid bilayer. We'll talk about that in a little bit. They are known as obligate intracellular parasites. So that means that they have to take over a cell and get inside of a cell in order for them to be able to reproduce, go through metabolism, all those types of things. So they kind of exist in this gray area where they are living but not living at the same time. They have DNA, they have genetic material, but at the same time they can't reproduce on their own. So they're kind of in this weird place. And they have to get into a cell in order to be able to perform their functions. These are some of the structures, not all inclusive of various viruses. We can have the tobacco mosaic virus, which has this kind of interesting capsid protein layer that kind of goes around the outside of the RNA, which kind of spirals on the inside. We can have protein capsid that has geometric shapes. In this case, I believe this is a dodecahedron. But there are many different types of shapes that they can have. Here we've got one that has the lipid layer, the membranous envelope. It's usually just called an envelope. The capsid is on the inside of the envelope, and the capsid then surrounds the RNA. That's an example of influenza. We can also have bacteriophages. Bacteriophages infect bacteria. They don't infect humans, and these kind of have this weird like space alien type thing, which is kind of cool. So in order for replication to occur, viruses must have a host cell, and we can have what is known as a host range, which is the range of species that a virus can infect. So viruses aren't necessarily tied down to just infecting one species. Many times they can infect, a couple different species that are generally closely related to one another, although at times they can also be, completely unrelated to one another, which is, kind of interesting. Specific viruses recognize cells based on the proteins that are located in the extracellular matrix. Those are like, proteins that signal that this is a nerve cell or a blood cell or whatnot. That's how viruses detect which cells they want to infect, or not necessarily want to infect, but which cells they are going to detect, because they have proteins around their capsid which are going to attach to the proteins in the extracellular matrix and kind of facilitate the binding. And again, different species of animals or different types of viruses, they all have different proteins. So that's all extremely important in terms of which viruses are going to be able to infect which species. This is just kind of a diagram showing you the general replication. What happens is that the virus will gain entry and uncoat, so the DNA or RNA needs to be kind of exposed inside the cell. Once it's inside the cell, the DNA will go through replication. The DNA can then be turned into RNA, which will then be used to produce more capsid proteins. They reassemble, and then the viruses exit out of the cell. So first thing we're going to do is take a look at some of the life cycles of some phages that infect bacteria. The first is called the Lytic Cycle, which is derived from the word lysis, which means to break open. And it does culminate in the death of the bacterial host cell. And what happens is that during the final stage, so that stage 4 that we were just looking at, instead of just kind of the viruses exiting the cells, they actually kind of break open, and the viruses are all released. And when it does that, it kills the bacterial cell. Now, bacteria do have defenses against these, which are include restriction enzymes. Classic example is restriction enzymes. And restriction enzymes are enzymes that will recognize a specific DNA sequence and cut it out. And generally, those DNA sequences are found in viruses instead of the own bacteria. Now, you might think that this would be really useful, right? These nice enzymes, they cut DNA. So restriction enzymes were, once they were discovered, were immediately kind of incorporated into a lot of the biotechnologies that we will be looking at as we kind of get into our biotechnology part of this unit. There is a constant evolution going on between bacteria and viruses, because you can imagine that all the viruses that get cut by the restriction enzymes they all don't survive. Now you're only left with viruses that aren't able to be, cut by the restriction enzymes, and so lots of evolution going on there. So this is kind of a diagram of the Lytic cycle. We've got our classic bacteriophage right here, T4, that has bound to the outside of the bacterial cell. It then injects its DNA and also degrades the host DNA. So it actually breaks down the host DNA and inserts its own. It then hijacks the molecular processes in order to build more viral DNA genomes and proteins. They are then assembled together, and eventually they break open, killing the cell and releasing all the viruses. We can also have the lysogenic cycle, and generally those viruses that go through the lysogenic cycle also go through the lytic cycle. And in the lysogenic cycle, rather than degrading the bacterial DNA, the phage DNA is actually incorporated into it. So it kind of gets inserted, and that's obviously really good because now as the bacteria is reproducing, whenever it duplicates its own DNA, it also duplicates the virus DNA. And so because of that, now the virus is being propagated, and at some point, some factors are going to trigger it to enter the lytic cycle. It will then produce viruses, be released, infect new cells. So let's take a look at animal viruses. Animal viruses are extremely varied. We can have either DNA or RNA as the genetic material, and they can either have or not have a viral envelope, or rather that kind of membrane that is derived from the host cell. And viral envelopes are heisted from a host cell, and they contain phospholipids and membrane proteins. So they actually, when they kind of are released out from the cell, they will steal some of the phospholipids. Too much variation in a typical life cycle for us to really kind of narrow it down and say this is the way a virus acts. They're all so different that there's no kind of like exemplar, but there are a couple of examples. One of the main examples that we're going to talk about is RNA viruses, and there are three main types of those. There are those that the viral genome acts as mRNA, and it's translated directly into proteins. We can have viral genomes acting as a template for the mRNA. So the RNA actually gets transcribed into other RNA, and then it uses an enzyme that it carries in the capsid to accomplish that. And then finally, we've got retroviruses, which are kind of the most well-known of these. And this is where viral RNA is turned back into DNA using enzymes called reverse transcriptase. So basically, we're doing the opposite of transcription, right? We're taking RNA, turning it back into DNA. The DNA is then incorporated into the host cell chromosomes, or the DNA is then used to create mRNA. HIV is an example. Ebola is another example, but there are obviously others that work a little differently. So let's talk about the life cycle of HIV, because it is such popular is not the right word, but it's one of those things that you're going to see a lot of over time, especially if you continue taking biology classes. They're going to talk about HIV a lot. So what happens is that we've got an envelope which has glycoproteins on the outside. The glycoproteins and the envelope kind of merge with the host cell. Reverse transcriptase is going to take the viral DNA. It is going to then produce an RNA-DNA hybrid. So you've got RNA with a complementary DNA. That RNA is then replaced with the DNA. The DNA is then incorporated into the chromosome. We call that now a provirus because it's not really a virus, but that DNA is in the cell. So we call it the provirus. That DNA is then read and produces more mRNA. The mRNA is then either used to produce proteins, the glycoproteins, which are or the capsids, which will reassemble to form new viruses, or that mRNA actually gets incorporated into the virus itself. And then the virus, again, it kind of takes some of the glycoproteins and the phospholipid bilayer and excretes itself kind of off in a process that's fairly similar to exocytosis, and then that's where we get our new virus. So how does actually getting HIV lead to AIDS? Well, HIV, which is human immunodeficiency virus, infects helper T cells, which are integral pieces in immune system response. And we will learn more about the immune system later on. But helper T cells are what facilitate the killer T cells being able to actually destroy incoming pathogens or things that are going to cause disease. So what happens is that over time, your immune system becomes unable to fight against things, which means that you now have AIDS, which is acquired immune deficiency syndrome. Now you're unable to fight off, basic infections that you would have if you're completely healthy. And eventually, you don't actually die directly because of HIV. You die because you get a cold or pneumonia or some other disease along the way. So kind of unfortunate in terms of how that kind of comes about. Now, how have viruses evolved, right? Because they're kind of like a weird thing. They're kind of in this semi-living state. And they're thought to have evolved from either of two things after cells. So cells evolve first and then viruses. They either could have evolved from plasmids, which are small circular DNA molecules that are found in bacteria. And these plasmids can be transmitted between bacterial cells, and they exist independently of the rest of the genome. Sounds kind of like a virus, right? Viruses are pieces of DNA or RNA that move around. We also think that they could come from transposons, which are mobile pieces of DNA. I know that sounds weird, but these are like pieces of DNA that can move from one part of your genome to another. And again, this is where the reason why this is thought to be is because it's DNA that's moving around, right? Just like a virus is kind of DNA that is moving around. And one of the reasons why we believe transposons in particular are good candidates for this is because viral DNA is usually has more in common. So it's usually is very similar to that of the host than it is to other viruses that infect the host. So like if we were to compare like rhinovirus, which does the cold, and influenza, rhinovirus and the cold would have more in common with us in terms of their DNA sequences than they would with each other. Okay, so kind of an interesting concept. But again, we don't know exactly which one it is yet. These are just kind of the main theories. Viruses are pathogens, which means that they are disease-causing agents. Viruses do most of their damage through killing or damaging cells that they infect. It's not that they really produce toxins that are released. It's just mostly the fact that they are infecting cells, and then those cells die or are damaged and can no longer do their function. Vaccines can prime the immune system to recognize viruses and stop infection, and they recognize the viruses based on the proteins that are on the outside of the virus. So viruses that have high mutation rates, it's hard to create a vaccine for because they're constantly changing the proteins that are on the outside of their capsid. So things like influenza and HIV are hard to develop vaccines for, right? Influenza, we have to develop a vaccine every single year because of the evolution that occurs along the way. HIV has one of the highest mutation rates, making it really difficult to develop a vaccine for. I think that was all. All right, lastly, we've got plant viruses. So viruses infect all living forms. Anything that has a cell, there is a virus for it, including plants. Most of them are RNA in terms of their genetic material, and they usually infect the host through an external source breaching the epidermis. So plants are particularly susceptible to viruses when they have like their epidermis, their leaves, bark, something like that, having been kind of corroded away. You can kind of see what it looks like when they have viruses. They usually get kind of blotchy and they don't look healthy, just like we tend to not look very healthy when we have the cold. So that's all for viruses. Thanks for watching, and I'll see you next time.